You know, we often talk about cancer using these grand military metaphors. It's an invasion, a battle, a hostile takeover.
Right.
But when you look at the research we have on the table today, specifically about breast cancer moving to the brain, it feels less like a war and, I don't know, more like a heist movie.
A heist movie is actually a surprisingly good analogy. I like that. You've got a specialist team, a very specific target that's supposed to be impenetrable.
The vault.
Exactly, the vault. And a really distinct set of tools they use to break in.
Right, because the brain isn't just another organ, is it? It's a fortress. Yeah. It has the blood-brain barrier, a totally different immune landscape, and, I mean, biologically, it is a very, very tough place for a foreign cell to find food.
And that is the central mystery we are unpacking today. We're looking at a study from Nature Genetics that, well, it essentially hands us the blueprints for this exact heist.
Yeah.
It explains the who, the genetic driver, the how, this incredible metabolic switch, and even the inside man, the environmental accomplice.
The paper is titled, and bear with me here, P53 inactivation drives breast cancer metastasis to the brain through SCD1 upregulation and increased fatty acid metabolism. Now, that is a mouthful.
It is, yeah.
But by the time we're done with this deep dive, I want every word of that title to feel like a plot point in the movie.
It really is a narrative,
yeah.
And it all starts with a very famous gene, P53.
Ah, the guardian of the genome. I feel like anyone who's read even a little pop science knows P53. It's the hero protein, right? If your DNA gets damaged, P53 steps in and says, hold on, stop dividing, fix this, or you die.
Correct. It's the ultimate tumor suppressor. But here's where the story takes a really sharp turn. The researchers didn't just find that P53 was mutated, which is, common in a lot of cancers.
Sure.
They found a statistical anomaly that is almost hard to believe. When they looked specifically at breast cancer brain metastasis, let's just call it BCBM for short, they found P53 inactivation in nearly every single sample.
Okay, I want to push back on that for a second because nearly every such a strong claim. In biology, we usually see correlations of 40%, maybe 60%. Biology is messy. Did they really find a link that consistent?
They did. In one specific cohort of these brain metastasis samples, the rate of TP53 perturbations, that's either a mutation or what's called a copy number loss, where the gene is just physically missing.
God.
It was 100%.
100%. That's not a correlation. That's a requirement. It suggests a bottleneck. It's not just that P53 loss makes the cancer aggressive. It implies you literally cannot survive in the brain without losing P53. It's like a cover charge to get into the club.
That is the implication. And to prove it wasn't just a coincidence, they moved from observation in humans to a causal experiment in mice. They used what are called isogenic cell lines.
And isogenic just means identical twins, basically.
Exactly. They took a line of breast cancer cells that had a perfectly functional P53. Then, using CRISPR, they just they deleted that one gene. So you have two cell lines, identical in every single way, except one has the guardian and one doesn't.
And when they injected them into the mice?
The difference was just night and day. The cells with normal P53, they rarely formed brain tumors. I think it was only 2 out of 11 mice, and the tumors were tiny.
Okay.
But the cells without P53, they colonized the brain in 12 out of 13 mice. Mm-hmm. And they formed these massive lesions.
So, okay. Recap number 1, the who in our heist is a cell that's lost its guardian, P53. And this isn't just a coincidence. Experiments show that losing P53 is what drives the ability to grow in the brain, but not necessarily other places. It's the genetic permission slip.
It's the key that gets you through the first door.
But why, this is where I always get stuck, P53 is a generic safety inspector. Why does firing the safety inspector suddenly give you the superpower to survive in the brain specifically? Why not the liver or the lungs?
And that brings us to the environment. The where. The brain is unique because of its lipid profile. We call it a lipid desert.
Which just sounds so contradictory. I mean, the brain is 60% fat. It's the fattiest organ in the body. How can it possibly be a lipid desert?
It's all about availability. Yes, the brain itself is full of structural lipids, myelin sheaths, cell membranes, but the space between the cells, the interstitial fluid where the cancer first lands, is incredibly low in free fatty acids.
So there are no snacks lying around.
There are no snacks. So the cancer cell lands in the brain and it needs to grow, which means it has to build new cell membranes. Cell membranes are made of fat, but there is no fat to eat.
Okay, so it can't import the building materials.
Precisely. So if you can't import fat, you have to do what?
You have to make it yourself.
You have to manufacture it. This is a process called fatty acid synthesis, or FAS. And this is where the P53 connection finally clicks into place.
Okay, let's try to visualize this. So the cell is a construction site. It needs bricks lipids to build its new walls. There are no bricks on the ground, so it needs to fire up its own brick factory.
And the form of that brick factory is an enzyme called SCD1, stearoyl-CoA desaturase 1.
A very catchy name. What does SCD1 actually do?
It's a converter. So the cell can make these very basic saturated fats, think palmitic acid and stearic acid. But you cannot build a functional cell membrane out of just saturated fats.
Because they're solid at body temperature, like butter.
Exactly. If a cell membrane has too many saturated fats, it becomes stiff, waxy, it loses its fluidity, signaling stops working, the cell basically chokes on its own fat and dies from something called lipotoxicity.
Okay.
You need monounsaturated fatty acids, MUFAs, to keep things fluid. Think olive oil.
So SCD1 is the machine that turns the hard butter into the fluid oil.
Yes. It just inserts a double bond into the fatty acid chain. It turns palmitic acid into palmitoleic acid and stearic acid into oleic acid. And this conversion is absolutely critical for the cancer cell's survival.
And P53's role in all this?
Well, in a healthy cell, P53 suppresses SCD1. It keeps that factory running a bare minimum because, you don't want to just overproduce fat.
Right.
But in our brain, metastasis cells, P53 is gone.
So the brakes are cut. The SCD1 factory goes into total overdrive.
A massive upregulation. The P53 null cells become these self-sufficient lipid generators. They don't need to find food in the desert. They brought their own power plant.
Okay, so let me just pause and map this out to make sure I've got the chain of events straight.
Go for it.
Step 1. A breast cancer cell loses its P53 gene. That's our genetic trigger.
Because P53 is gone, it can't hold back the SCD1 enzyme anymore.
SCD1 levels just skyrocket. This is the metabolic engine.
And this engine lets the cell make its own liquid fats, the MUFAs, which lets it survive and build in the lipid desert of the brain. Is that the whole picture?
It's the core of it, but it's missing the accomplice, the inside man. Because even a factory needs raw materials. You can't make lipids out of thin air. You still need carbon sources.
And this is where the story gets a little disturbing, because the cancer cells aren't just ignoring the brain tissue, they're actively exploiting it. Enter the astrocytes.
Astrocytes are fascinating cells. They're these star-shaped glial cells that normally support neurons, keep things tidy, but in the context of a tumor, they get activated. And the paper shows how these astrocytes start secreting all these factors into the environment.
They're feeding the tumor. Unwittingly.
Yes. Specifically, they're pumping out things like glutamate and other metabolic primers that the cancer cell can use to fuel that SCD1 factory. But the cancer cell needs a way to get those nutrients inside. It needs a door. And
let me guess, P53 controls the door too.
It does. The door is a transporter protein called CD36. And the study found that when you lose P53, it leads to a huge upregulation of CD36.
Oh, that is diabolical. So losing P53 does two things at the same time. It opens the delivery bay doors at CD36 to suck up resources from its neighbors. And it turbocharges the factory SCD1 to process those exact resources into building materials.
It creates a perfect, deadly metabolic storm. And the visual evidence in the paper is striking. They use these fluorescent stains to look at the tumors, and you can literally see the cancer cells, which are expressing high CD36, physically clustering right next to the astrocytes.
They're cuddling up to the food supply.
It's a parasitic relationship. The astrocyte thinks it's helping out a damaged cell, but it's actually fueling the invasion.
So let's do our second recap, the how. The cancer survives because the brain is a lipid desert, so it has to build its own fat. The key enzyme for this is SCD1. Normally, P53 keeps SCD1 in check, but with P53 gone, SCD1 runs wild. And to top it off, local brain cells, astrocytes, unwittingly feed the cancer the raw materials it needs.
That's the complete picture of the heist.
But for the listeners who really want to get into the machinery, because that's where the potential drugs come from, we need to peel back one more layer. We said P53 stops SCD1, but P53 works on DNA, right? It doesn't just physically hold the enzyme back. There has to be a middleman.
This is the deep mechanism part of the paper, and you're right, it's crucial for drug development. The researchers found this protein cascade. It involves two other acronyms you need to know: SREBP1 and DBT1.
Okay, let's take SREBP1 first. It sounds like a droid from Star Wars.
Yeah, it kind of does. SREBP1 is the master switch for making fat. When this protein gets into the nucleus, it binds to the DNA and turns on a whole suite of genes related to making lipids, including our main character, SCD1.
Okay, so SREBP1 is what actually turns the factory on. What's DPDC1?
UTPC1 is the co-activator. Think of it as SREBP1's manager. SRBP1 can't really do the job effectively on its own. DPDC1 comes along, binds to SREBP1, and stabilizes it. It helps it stick to the DNA and do its job without getting broken down.
Okay, so let's walk through the chain of command then. Yeah. In a healthy cell, the CEO, P53, is in the office. The CEO tells the middle manager, DPTT1, to take a hike.
Right.
And without the middle manager there to help, the foreman, SREBP1, is lazy and doesn't really turn on the factory, SCD1.
That's a great way to put it. Now, in the cancer cell,
the CEO, P53, is missing. So the middle manager, DEPDC1, comes back and starts shouting orders. He grabs the foreman, SREBP1, and together they force the factory, SCD1, to run 24/7.
Exactly. And they had hard evidence of this physical interaction. They use a technique called chick-IP. Essentially, they freeze the proteins that are attached to the DNA and take a snapshot. They showed DDC1 and SREBP1 were physically sitting on the promoter region of the SCD1 gene.
The smoking gun.
It's the smoking gun.
And this detail matters so much because if you want to stop the cancer, you have to know which part of the machine to break. If you can't put the CEO back in charge, maybe you can sabotage the factory itself.
Exactly. And that leads us to the therapeutic hope in the paper. The researchers realized that these brain metastasis cells are, for all intents and purposes, addicted to this pathway.
Addiction implies vulnerability. If I stop eating fat, well, I'll be fine because my body has other ways to get energy. But these cells,
they cannot adapt. They have rewired their entire existence around this SCD1 factory. If you block it, they hit a brick wall. They start accumulating all those toxic saturated fats, their membranes harden, and they die.
So, what drugs do we have? The paper mentions a couple, SW203668 and A939572.
These are specific small molecule inhibitors of SCD1. And the testing process they used here was very, very rigorous. They didn't just stop at cells in a petri dish.
Right. They used those 3D spheroids first, the little mini tumors, and saw the P53 null cells dying off. But the mouse data was what really caught my eye.
The mouse data was significant. They treated mice that already had established brain metastases, and the drug, SW203668, was able to cross the blood-brain barrier, which is a huge hurdle for most cancer drugs.
That's a huge deal.
And it significantly shrank the tumors.
But here's the key question. Did it kill the mouse? Because if you stop fat production in the whole body, that sounds dangerous.
That is the beauty of the addiction concept. The normal cells in the mouse, they have functional P53. Their lipid metabolism is flexible, they can handle it. The cancer cells, however, had no backup plan. So the toxicity was highly selective to the tumor.
And they went one step further, didn't they? With human tissue.
Yes, patient-derived organotypic cultures. This is really the gold standard before you even think about a clinical trial. They take slices of brain metastasis that have been removed from actual patients during surgery.
So it's a real slice of the disease environment and all.
Exactly. And when they applied the SCD1 inhibitor to these human tissue slices, they saw significant apoptosis. That's programmed cell death in the tumor cells, while the surrounding healthy brain tissue remained largely viable.
That is incredibly promising. It takes this whole story from cool biology trick to potential medicine.
It validates the target. It tells us that this mechanism isn't just something weird that happens in mice, it is actively driving the disease in humans.
Okay, so let's do a final recap, the solution. We've unraveled this whole protein chain. P53 normally suppresses DEPDC1, which stops SREBP1 from activating SCD1. In these tumors, that chain is broken, and they become addicted to SCD1. So the solution is to block SCD1 with drugs, which successfully shrinks tumors in mice and in human tissue samples.
It's a targeted attack on the factory the cancer built to survive.
So this really moves us toward a future where we don't just treat breast cancer, we profile it. If a patient comes in and their primary tumor shows a loss of P53 and maybe high levels of this DEPDC1 protein.
And we know they are at a very high risk for brain metastasis, and we know exactly why. They are metabolically primed to survive in that lipid desert.
And potentially, we could treat them with an SCD1 inhibitor.
That's the goal.
But this raises a provocative question, and I want to get your take on this. Traditionally, we wait. We wait for metastasis to show up. We do an MRI, we see a spot, and then we treat it. But if we know the mechanism, if we have the blueprints to the heist before they even enter the bank, should we be treating prophylactically?
That is the billion-dollar question in oncology right now.
I mean, if you see the P53 loss in the primary breast tumor, you know the cancer has the visa for the brain. Why wait for them to colonize? Could we give an SCD1 inhibitor to effectively starve these potential invaders before they can ever establish a beachhead.
Biologically, it makes perfect sense. If you make the brain environment inhospitable by blocking the one enzyme they absolutely need to survive there, then they might arrive, but they won't thrive. They'll die on arrival.
But clinically, that's a whole other ballgame.
Clinically, it's harder. Giving a systemic drug to someone who might get metastasis is always a risk. You have to balance the potential side effects against the potential benefits. But studies like this are what shift that paradigm. If the correlation is as strong as this paper suggests, that 100% link, then the risk profile changes. The maybe suddenly becomes a probably.
It shifts from being a gamble to being a calculation.
Exactly. And it really emphasizes that we need to stop thinking of cancer as this static lump. It's an evolving system. It changes its metabolism to match its zip code. Our treatments have to be just as adaptable.
It's a fascinating, a little terrifying.
Right.
Look at how adaptable life, even corrupted life like cancer, can be. But the fact that we can trace the steps all the way from a missing gene to a rogue enzyme, that gives us the roadmap to stop it.
Knowledge is the first step to intervention. We know the route they take now. We just need to set up the roadblock.
For those of you who want to see that factory schematic we were talking about, that whole chain from P53 down to SED1, we've put the key diagrams from the paper in the show notes. It really helps to see the hierarchy visually.
I'd also highly recommend looking at the immunofluorescence images of the astrocytes. Seeing the cancer cells literally huddling around them really drives that accomplice point home.
Definitely check those out. Thanks for taking this deep dive with us. It's complex science, but it could end up saving lives.
Always a pleasure to explore it.
We'll see you on the next deep dive.